Welcome to the deep dive. Today, we are taking, well, the ultimate field trip, I suppose.
Yeah.
Navigating the cytoplasm, slipping past the membrane,
Right.
and heading straight into the cell's command center, the nucleus. We are here to meet the ultimate executives.
Right. The real decision makers.
Exactly. The proteins that take, a fleeting biological whisper and translate it into something durable, something physical, a real change.
Yeah, it's a difference between hearing a rumor and seeing a whole new skyscraper actually get built. We spend so much time talking about signals.
Right. Growth factors, hormones.
Keratin, endocrine, all that, things hitting receptors. But those are just the incoming memos, like you said.
Just a message.
Just the message. What we're looking at today is the foundational process. How does that message grow now, or maybe stop working, differentiate? How does that get converted into a lasting change? In the cell's architecture, its identity, its behavior.
So we're dealing with the master architects then, these ultimate control mechanisms.
Yes.
The transcription factors, or TFs.
That's them. These specialized protein complexes, they sit right at the absolute end of, well, almost every signal transduction pathway you can think of.
They're the executors.
They are the executors, absolutely. If a cell is going to change its function, divide, specialize, or even initiate its own programmed death.
Apoptosis.
Right, apoptosis. It does so by changing the genes it expresses. And TFs. They're the ones responsible for modulating that gene transcription. They drive those durable physical effects we talked about.
And it's obvious how critical this is, because when these factors go wrong, when they're mutated or regulated improperly, the consequences just ripple outwards across the entire organism.
Oh, completely. We're talking about fundamental players here, things like MYC and JUN.
Yeah, the pro-growth ones.
Exactly, pushing the cell towards rapid proliferation.
Yeah.
And then the flip side, like TP53, the famous tumor suppressor.
The guardian of the genome, they call it.
That's the one, a cell cycle inhibitor. And these TFs, they don't just, tweak one little gene here or there. They have massive widespread effects. They're rewriting entire chapters of the cellular script.
So if you really want to understand, say, cancer or development or even how the immune system works,
you have to understand transcription factors, how they're activated, how they're regulated, all the intricate details. They are arguably the single most important control point in cell biology.
Okay, let's unpack this then. If they're that critical, how do these master switches actually get thrown? What are the actual mechanisms that let them get access to the DNA in the first place?
Right. So first, let's just unify the signaling landscape a bit. It doesn't really matter where the initial signal comes from, whether it's local paracrine or a hormone from faraway endocrine, or even a nerve signal synaptic. The ultimate goal very often for the cell is to activate a transcription factor and get it physically into the nucleus.
Okay, activation and location.
Activation and nuclear localization, yeah.
And the path they take, it's not about the signal's origin, you said. It's about the signal molecule itself.
Right.
Like, can it get through the front door, the cell membrane?
Precisely. That's the key difference. Can it cross the cell membrane? So we end up with two main routes based on the physicochemical properties of the ligand, the signaling molecule itself.
Okay, route one, intracellular receptors. Sounds like the simple one, the express lane.
It is, relatively speaking. This direct route is for lipid-soluble ligands, things that like fats.
Okay, like what?
Think about crucial molecules like steroid hormones, testosterone, estrogen, cortisol, or vitamin D. Because these molecules are nonpolar, hydrophobic.
They like water.
Right, they don't like water, they do like lipids. So they can easily just dissolve right through the fatty plasma membrane, slip right into the cytoplasm, no need for a fancy receptor on the outside.
So the signal doesn't need a middleman, it just strolls right in and finds its target inside.
Correct. And once inside, these lipid-soluble ligands bind directly to their specific partners, the nuclear hormone receptors. And here's the key insight for this route. These nuclear hormone receptors are the transcription factors themselves.
Ah, okay, so the receptor is the TF.
Exactly. They're often just hanging out, maybe in an inactive state, sometimes in the cytoplasm, sometimes already in the nucleus, perhaps bound to inhibitory proteins that keep them quiet.
And when the ligand, the hormone, binds, what happens?
Binding the ligand causes a conformational change, a shape shift in that receptor TF complex. This often kicks off those inhibitory chaperone proteins.
Lets them go.
Lets them go. And now you have this stable receptor ligand complex, which is the active TF.
Yeah.
It then directly binds to specific regulatory sequences on the nuclear DNA, and boom, it activates or maybe represses gene transcription.
Wow, so it's incredibly fast. One step to the nucleus, basically. Allows for quick hormonal responses.
Super quick, super powerful. It's a really elegant system.
Beautifully simple. But you mentioned most signals don't get this easy ride.
No, sadly not. The vast majority of signaling molecules, think peptides, proteins, neurotransmitters, they're just too large or too polar, too charged. They simply cannot cross that lipid barrier on their own.
So they have to use route 2, the complex relay race. They need those cell surface receptors.
That's it. The signal stops at the door, essentially, and has to hand off the message.
Okay.
So these cell surface receptors are typically transmembrane proteins. They span the membrane. When the ligand binds to the part sticking outside the cell...
External domain.
Right, the external domain. That binding causes a critical change in the receptor's shape, its conformation. Often, this leads to receptors clustering together. They might dimerize, two coming together, or form larger groups.
And that physical change, the clustering or the shape shift, that's what starts the whole thing off inside the cell.
It's the starting pistol for the relay race, exactly. It initiates this whole sophisticated intracellular cascade. We call it a signal transduction pathway. And almost always, it relies heavily on adding or removing phosphate groups, phosphorylation and dephosphorylation, over and over.
So if the signal molecule, the ligand, can't get to the nucleus itself, this whole cascade, this relay race, its job is to activate a TF that's already inside, or get one inside.
That is the entire objective. The cascade aims to wake up a latent, sleeping transcription factor. And it can do this in several ways. The source is mentioned triggering maybe a proteolytic event where the TF is cut to become active.
Like the Notch pathway we'll maybe discuss later.
Exactly like Notch. Or maybe the cascade changes the TF's overall stability, stops it from being destroyed so quickly.
Right.
Or, probably most commonly, it modifies the TF chemically, usually through phosphorylation, to change its location, physically shuttle it from the cytoplasm into the nucleus where all the DNA action is.
Okay, so phosphorylation seems absolutely central here. It's the core mechanic, the internal language of this relay race. Let's get into the nitty-gritty of how that works. How does adding a phosphate group actually pass the message along?
Phosphorylation is the critical switch. It really is. Take a receptor tyrosine kinase, an RTK, for example. When the ligand binds outside...
Like a growth factor.
Like a growth factor, yeah. The receptors cluster, and this new shape, this new geometry, it stimulates the kinase activity that's built right into the receptor's own tail, inside the cell.
So it activates itself.
Often, yes. It leads to autophosphorylation, where the receptor adds phosphate groups to itself, specifically onto tyrosine amino acids on its own tail. It becomes like a docking platform.
A signaling platform, right?
Yeah.
Ready for other things to bind.
Precisely. Now, in other cases, for receptors that don't have their own built-in enzyme activity, think some immune receptors or integrins that sense the extracellular matrix ligand binding doesn't activate the receptor itself, but it encourages recruited, separate enzymes called non-receptor tyrosine kinases to get active. The Succic family kinases are probably the most famous example.
So whether it's self-activation or recruiting help, the result is the same. Adding that charged phosphate group, that's the on switch, done by enzymes called kinases.
Kinases add these negatively charged phosphate groups, usually to tyrosine, serine, or threonine residues on the target protein, and that simple addition drastically changes the protein's structure, its charge, its function. But, and this is really crucial, it's not a one-way street.
Right. You need a way to turn it off, too.
Absolutely. We have to give equal credit to the phosphatases.
The counterpoint. So if kinases put the phosphate on, phosphatases take it off.
Exactly. If kinases are the gas pedal, phosphatases are the brakes, or maybe even the reset button. They remove those phosphate groups, typically inhibiting the signal, restoring the protein to its original state. This allows the pathway to switch off quickly and be ready for the next signal.
So it's this dynamic balance, this tug of war between kinases and phosphatases that controls how long and how strong the signal response is.
It dictates the precise duration and intensity. It's incredibly fine-tuned.
Now, the traditional view, the one maybe we learned in introductory biology, was very linear. A activates B, B activates C, C activates the TF. Like dominoes falling.
Right, the simple chain.
But modern biology, especially with systems biology and computational approaches, sees it very differently, doesn't it? More like networks, nodes, and hubs.
That shift in perspective is absolutely critical. The reality is far more intricate. That initial signal hitting the receptor doesn't just trigger one single pathway. It often explodes outwards, impacting multiple processes simultaneously.
Wow.
So this idea of modular signaling emerges. A single receptor activation might, at the same time, influence cell division and tweak metabolism and reorganize the cell's internal skeleton. These nodes you mentioned are key proteins where signals converge or split off, allowing for integration and crosstalk between pathways.
Okay, that sounds incredibly complex, like a massive interconnected switchboard. How did the cell keep everything straight? How does it prevent signals from, accidentally jumping tracks or causing chaos? How does it ensure specificity?
That's where the adapter proteins come in. They are the molecular connectors, the scaffolding elements. These proteins are fascinating because they don't usually have any enzyme activity themselves.
They don't do anything chemically.
Not directly, no, but they are essential for linking other enzymes together and assembling the specific signaling complexes that define those hubs we talked about. They're the organizers.
They dictate the wiring diagram of the network.
Mm.
Can you give us a specific example of how they enforce that specificity? They must recognize exactly the right partner.
Okay, a classic example involves specific functional regions or domains found on these adapter proteins. Let's talk about the SRC homology domains, or SH domains. The SH2 domain is perfect.
Okay.
An SH2 domain is like a molecular hook. It's specifically designed to bind to a protein, like an activated receptor, that has been phosphorylated on a tyrosine residue.
So it recognizes the phosphate tag.
It recognizes the phosphotyrosine, yes. But here's the beautiful specificity part. The SH2 domain doesn't just see the phosphotyrosine. It also recognizes the specific sequence of, say, 3 or 4 amino acids immediately surrounding that phosphorylated tyrosine.
So it's not just seeing the phosphate, it's reading the local address, too. It's like a molecular postcode.
Exactly. A molecular postcode. That's a great analogy. This incredible specificity ensures that only the correct adapter protein docks at that specific activated site on the receptor or another signaling protein.
Preventing the wrong connections.
Absolutely. It allows the cell to bring together the right subset of enzymes and regulatory proteins, the specific components needed for that particular signal's hub right there at the signaling site. It ensures fidelity. And then you have other domains, like the SH3 domain, which often binds to proline-rich sequences on other proteins, recruiting yet more components to build the functional complex before the final instruction gets passed down the line towards the TF. This modularity, using these specific domains, is key to precise signaling. Hashtag TAC 2.3, the anatomy of a transcription factor.
Okay, so we followed the signal from the outside, maybe through that complex relay race with adapters and phosphorylation, and now finally, the command has reached the transcription factor itself. You mentioned earlier they're complex. What's the structure? What allows them to interact with both the incoming signal and the DNA?
Right. TFs aren't just, simple blobs. They are definitely complex, and crucially, they're built in a modular way, like a Swiss Army knife, maybe, with different tools for different jobs.
Okay, different functional parts.
Exactly. This modular design is what allows them to be regulated by all those upstream signals we just talked about, while still keeping their core ability to bind DNA and recruit the necessary machinery.
So what are the essential modules, the key tools in the TF toolkit?
Well, broadly, we can define 3 primary functional modules or domains. First, and absolutely non-negotiable, is the DNA binding domain, the DBD.
Makes sense. It has to bind DNA.
It has to bind DNA. This domain allows specific high-affinity binding to short, precise DNA sequences, those genetic on-off switches. Without a functional DBD, it's just not a transcription factor. It can't do its main job.
Okay, module 1, DNA binding. What's number 2? It relates back to that first route, the simple one.
It does indeed. The second is often called the small molecule domain or ligand binding domain. These are specialized pockets designed to bind directly to specific small signaling molecules, like those steroid hormones we mentioned earlier.
So the nuclear receptors have this domain.
Exactly. This module is what allows them to be directly activated by their ligand, bypassing the whole surface receptor cascade. It's a defining feature of that class of TFs.
Okay. And the third domain must be the connection point for route 2, the relay race.
That's right. You can think of it as the intracellular regulatory protein domain, or sometimes just the activation domain. This is the interaction zone. This is the surface that binds to those kinases, phosphatases, adapters, and other signaling proteins involved in the cascade.
So this is where the phosphorylation signal, for instance, actually lands on the TF.
Precisely. This domain receives the crucial regulatory input from the upstream pathway.
And receiving that signal, that phosphorylation or other modification, this leads us to what are called post-translational modifications, or PTMs. You mentioned these like the final check, the executive access pass for the TF.
That's a great way to put it. PTMs are the ultimate fine-tuners, the controllers of TF function after the protein has been made. These modifications, especially phosphorylation, but also things like ubiquitination or acetylation, dictate what the TF actually does next. They control at least 4 essential aspects of TF function.
4 key controls. What's the first one?
The first critical control is translocation. Moving house. Often the TF is made and kept inactive in the cytoplasm. A PTM event, like phosphorylation, can expose a nuclear localization signal, basically a ticket into the nucleus.
So the PTM tells it, okay, time to go to the nucleus where the DNA is.
Exactly. Or conversely, a PTM might cause it to be exported out of the nucleus. Controlling its location is fundamental. A TF stuck outside the nucleus when it needs to be inside is useless.
Okay, control 1, location. What's second?
Second is controlling its lifespan. Protein half-life. PTMs, particularly ubiquitination, can tag a TF for destruction by the cell's garbage disposal, the proteasome.
Making the signal short-lived.
Making it transient, yes. Or other PTMs can stabilize the TF, protecting it from degradation. This makes the signal and the cellular response much more prolonged and durable. It's a really powerful regulatory lever. How long does the TF stick around?
Okay, location, lifespan. Third, this must involve the DNA itself.
Oh, yes. The third control is DNA binding ability. A PTM can directly alter the TS shape, maybe unmasking the DNA binding domain that was previously hidden or blocked by an inhibitory protein.
Like taking off a safety cap.
Sort of like that, yeah. Or it might enhance the affinity of the DBD for its target DNA sequence. It acts like a key turning in a lock, ensuring the TF binds effectively to the right genetic sequence, but only when the signal says so.
Okay, location, lifespan, DNA binding. And finally, the fourth control, that this is the payoff, right? Getting the job done.
This is the ultimate objective. RNA polymerase recruitment. A TF doesn't actually transcribe the gene itself. The heavy lifting is done by a huge enzyme complex called RNA polymerase.
The TF is just the guide.
It's the recruiter and the guide. PTMs on the TF, often within that activation domain, are crucial for attracting and binding RNA polymerase and its associated factors, called general transcription factors, to the gene's starting point. The TF essentially tells the polymerase, start here and transcribe this gene actively. It augments transcription activity. #tag 2.4 gene regulation and chromatin organization.
So the activated TF, armed with its PTMs, gets into the nucleus, can bind DNA. Now, where does it bind? I remember reading that it's not always simple, not always right next door to the gene it controls.
No, that's one of the really fascinating and initially quite confusing aspects of gene regulation. Some TFs do bind in what we call promoters. These are DNA sequences located very close to the transcription start site of a gene, maybe just upstream. That's the classic kind of intuitive model.
The simple case.
The simple case. But for coordinating complex processes, think building an entire organ during development, or shifting the cell's entire metabolism, you need to control lots of genes at once in a coordinated fashion.
Right, a whole program needs to run.
Exactly. So many sophisticated TFs don't just bind to one promoter. They might bind to regulatory sites scattered across the entire genome, simultaneously activating or repressing a whole cassette or regulon of dozens, maybe hundreds of interrelated genes needed for that complex change.
Okay, binding all over the place.
Yeah.
And this brings us to something you hinted at, the idea that location isn't just about the linear sequence of DNA letters, but the actual 3D structure. This is where enhancers come in, right?
Precisely. This is a huge aha moment in understanding modern genetics. Enhancers are regulatory DNA sequences that can be incredibly far away from the gene they control in terms of the linear DNA sequence.
How far?
Tens of thousands, even hundreds of thousands of base pairs away. They might be upstream, downstream, even located within an intron, a non-coding section, of a completely different neighboring gene.
Wow. So how on earth can a piece of DNA so far away physically influence the transcription of a specific gene way over there? It sounds like action at a distance.
It looks like action at a distance, but it's actually about bringing things together in 3D space. It's not magic, it's clever molecular engineering. Enhancers work by binding specific transcription factors and other regulatory proteins.
Right.
And this collection of proteins bound to the enhancer then physically interacts with the promoter region of the target gene, even though they're far apart on the linear strand. They do this by causing the DNA itself to loop out, bending the massive DNA molecule.
So it forms a loop, bringing the distant enhancer physically close to the gene's starting point.
Exactly. They become geographical neighbors within the packed environment of the nucleus, even if they're miles apart on the linear map. This looping brings the activating proteins recruited by the enhancer right next to the gene's promoter, dramatically boosting the recruitment of RNA polymerase.
That's incredible.
Yeah.
It really underscores that DNA isn't just a static string of code. It's a dynamic 3D structure constantly being folded, looped, and reorganized inside the nucleus.
Absolutely. It emphasizes the fundamental importance of chromatin organization. Chromatin is the complex of DNA and proteins, mostly histones, that forms chromosomes. How that chromatin is structured, whether it's tightly packed and inaccessible or open and loose, is a major layer of gene regulation. The TF isn't just reading the code, it's often acting like an architect, reshaping the physical structure around the code to make it accessible.
And to do that reshaping, that bending and looping, the PF can't do it alone. It needs help, right?
Yeah.
Like a construction crew.
It absolutely needs a team. Those protein interaction domains on the TF, the ones that receive the PTMs, are key here. They serve as recruitment platforms for this auxiliary crew.
Okay, who's on the team?
We're talking about several types of crucial helpers. Coactivators are proteins that bridge the TF to the general transcription machinery. Then you have histone-modifying enzymes.
Histones are the proteins DNA wraps around, like spools.
Exactly. DNA is wrapped around histone proteins to form structures called nucleosomes. If the DNA is wrapped too tightly, the transcriptional machinery, like RNA polymerase, literally can't get access to the gene's promoter.
So the histone modifiers loosen things up?
Precisely. Enzymes that add acetyl groups, histone acetyltransferases, or HATs, are often recruited by activator TFs. Acetylation tends to neutralize the positive charge on histones, loosening their grip on the negatively charged DNA. This opens up the chromatin structure,
Making the DNA accessible.
making it accessible. And then you also have chromatin remodeling complexes. These are large multi-protein machines that use energy, usually from ATP, to physically slide or even evict those histone spool nucleosomes out of the way, clearing the promoter region for RNA polymerase.
So the TF is like the foreman. It binds the site, then calls in the histone modifiers to loosen the structure, the remodelers to clear the path.
And the coactivators to help flag down the main machinery. It orchestrates the whole site preparation.
All leading up to the final, most critical recruitment.
The arrival of the star player, the RNA polymerase complex itself, along with its general transcription factors. The TF's successful recruitment and organization of its auxiliary team ensures that polymerase binds effectively, initiates transcription, and often stays active, leading to robust and sustained gene expression.
We've covered the main routes, direct binding for lipid-soluble signals and the phosphorylation cascade for others. But biology loves variations on a theme. Let's look at a couple of really interesting specialized pathways often used in development that have unique ways of activating their TFs.
Sure, we can quickly revisit the nuclear receptors just as our baseline, remember. The steroid hormone diffuses in, finds its receptor, which is the TF, and the activated complex goes straight to the DNA. Simple, direct.
Okay, the baseline. Now let's contrast that sharply with the Notch family receptors. The mechanism here sounds almost destructive. Proteolytic cleavage, cutting the protein.
It is quite dramatic. The Notch pathway is fascinating because it's crucial for direct cell-to-cell communication, often determining cell fate choices between adjacent cells. Here's how it works. A ligand protein sticking out from one cell surface binds directly to the Notch receptor protein sticking out from its neighbor.
A cellular handshake.
A very specific handshake. And this binding event, this physical interaction, apparently generates enough mechanical stress or conformational change in the Notch receptor that it triggers a rapid, irreversible cut proteolytic cleavage right near where the receptor passes through the cell membrane.
So the receptor protein literally gets snipped in two. Right. And one piece becomes the signal.
Exactly. The part that's released into the cytoplasm, the intracellular domain of Notch, often called NICD or IC Notch, is the newly activated transcription factor.
Wow.
It immediately detaches from the membrane, zips off to the nucleus, finds partner DNA binding proteins already there, and together they form a complex that regulates target gene expression.
That is incredibly direct. Mechanical interaction outside leads to a TF fragment inside, going straight to the nucleus. Very efficient.
Extremely efficient. A remarkable way to convert a direct cell contact into a nuclear command in basically 2 steps. Cut and translocate.
Okay, that's cleavage. Now let's look at another crucial developmental pathway, the Wnt-Frizzled pathway. This one uses a completely different strategy, doesn't it? More about Stabilization, preventing destruction.
That's right. The canonical Wnt pathway, canonical meaning the most well-studied version, is a master regulator of, again, cell fate, proliferation, tissue patterning. And its entire mechanism hinges on controlling the levels of its key transcription factor, a protein called beta-catenin.
Beta-catenin. Okay, so what's the default state? If there's no Wnt signal around, what's happening to beta-catenin?
In the normal unstimulated state, the default the cell is actively and constantly working to destroy beta-catenin.
Destroy it. Why?
Because beta-catenin is potentially very powerful, so it's kept on a very short leash. It's held within a large multi-protein scaffold called the destruction complex. Key components are proteins like axin, APC, and kinases like GSK3.
Okay.
Within this complex, beta-catenin gets phosphorylated by GSK3, and that phosphorylation acts as a tag, marking it for ubiquitination, the addition of ubiquitin chains.
And ubiquitin is the tag for?
The tag for the proteasome, the cell's garbage disposal. So tagged beta-catenin is immediately sent off and degraded. Its half-life in this state is incredibly short, maybe just minutes. It's constantly being made and just as constantly being destroyed.
Wow. So the cell is basically running a furnace, constantly burning up this potential TF. The Wnt signal then, it must put out the fire. It's not about making beta-catenin. It's about saving it from the fire.
Exactly. That's the core logic. When the Wnt protein ligand binds to its receptor complex on the cell surface, which includes frizzled and LRP56, this binding event recruits another crucial intracellular protein called disheveled to the receptor complex at the membrane. And disheveled's main job here is to interfere with and ultimately dismantle that destruction complex.
It breaks up the scaffold holding beta-catenin.
It disrupts the destruction complex, pulling key components like axin and GSK3 away from beta-catenin.
So beta-catenin is no longer being phosphorylated and tagged for destruction.
Correct. The result is immediate. Stabilized beta-catenin, no longer being sent to the proteasome furnace, rapidly starts to accumulate in the cytoplasm. Its concentration builds up.
And once it builds up,
it's free to translocate into the nucleus. There, it partners up with other DNA binding factors like TCF-LEF family proteins, and this new complex activates the transcription of crucial Wnt target genes, often involved in proliferation and development.
That's fascinating. It really highlights how controlling protein stability, that protein half-life PTM consequence we talked about, can be a primary mechanism for regulating a TF. It's all about preventing its destruction. Okay, this is all incredibly elegant, these control systems. But here's where it gets really interesting and maybe a bit scary. Because this intricate cellular control, especially the signaling pathways leading to TFs, this is precisely what goes haywire in so many diseases.
Absolutely. Unregulated transcription factor activity is right at the heart of countless proliferative disorders, and particularly cancer.
It's the engine of malignancy.
You could definitely see it that way. Think about those growth factor signaling pathways we discussed, the ones involving RTKs and cascades like resveratrol. The genes encoding many components of these pathways are what we call proto-oncogenes.
Meaning they're normally involved in controlled growth, but have the potential to cause cancer if something goes wrong.
Exactly. A mutation, maybe an amplification where the cell makes too many copies of the gene. This can convert a proto-oncogene into an oncogene. And an oncogene leads to a hyperactive pathway, one that's constantly screaming, grow, divide, even when it shouldn't be.
And the final output of that malfunctioning, screaming pathway is often a transcription factor that's stuck in the on position.
Precisely. TFs like MYC and JUN, which we mentioned, are normally involved in driving growth. They are very often the end targets of these pathways that get mutated in cancer. If you have a mutated RAS protein, for example, that's stuck on, it constantly signals downstream, ultimately leading to hyperactivation of TFs like MYC. The cell gets this relentless push to proliferate, completely ignoring normal external cues or internal checkpoints.
And when these pro-proliferation TFs like MYC get that unstoppable green light, they don't just, press the accelerator. They seem to do everything needed for division. It's like they hotwire the whole car. What are the key things they activate genes for?
It's really a comprehensive takeover strategy. They activate genes that coordinate at least 4 crucial activities needed for relentless cell division, all at the same time.
4 things, okay.
1, they promote entry into the cell cycle. They push the cell out of its resting state, G0, and force it into the active growth and division cycle. They override the initial gatekeepers.
Okay, get the cycle started.
2, they relieve blocks on cell cycle progression. They help the cell blow past the normal checkpoints that are meant to pause the cycle if something's wrong, like DNA damage, or if resources are low.
Ignore the warning lights.
Pretty much. 3, they actively prevent apoptosis. They turn on survival genes and turn off death genes, overriding the cell's own self-destruct program. This is crucial for cancer cells, allowing them to survive despite stress and accumulating mutations.
Keep dividing even when damaged.
And 4, they enhance the synthesis of building blocks. They ramp up metabolic pathways to crank out all the necessary raw materials, lipids for membranes, nucleic acids for DNA replication, proteins for everything else needed to physically build 2 daughter cells from 1.
Wow. That is a complete package for uncontrolled growth. Start the engine, disable the brakes, ensure survival, and fill the tank with fuel and materials.
It's a highly effective and dangerous strategy when unregulated.
But, as we also discussed, the cell isn't defenseless. It has built-in safety systems, the famous counterbalance to MYC and JUN.
TP53.
Ah yes, P53, the guardian. It's the perfect counterexample. TP53 is itself a transcription factor, but it's produced by a classic tumor suppressor gene. When activated, often by stress like DNA damage, P53 turns on genes that halt the cell cycle or initiate DNA repair, or if the damage is too severe, trigger apoptosis.
So it's the emergency brake. It's the emergency brake and potentially the self-destruct button. The delicate balance between these pro-proliferation TFs like MYC and JUN and the cell cycle arrest or death-promoting TFs like TP53 is absolutely critical for normal tissue health for homeostasis.
And when TP53 itself gets mutated, which happens in, something like half of all human cancers?
It's incredibly common. When you lose functional P53, that essential safety brake is gone. Cells accumulate mutations much faster, they don't die when they should, and they become much more prone to aggressive, uncontrolled growth. Losing P53 is a major step towards malignancy.
Okay, so given that these TFs, MYC, JUN, TP53, and many others are so central, sitting right at the control panel for cell fate, why don't we have more drugs that just directly target these TFs in diseases like cancer? It seems like the obvious place to intervene. Why do so many cancer drugs target things further upstream, like the receptors, the RTKs, or kinases in the cascade?
That is a really critical therapeutic question, and the answer gets into the practical challenges of drug development. You're right, targeting the upstream components has been very successful. The source material mentions drugs targeting growth factor receptors like EGFR or HAR2, which are often overexpressed or mutated in lung, breast, and brain cancers.
Right. Antibodies like Herceptin or small molecule kinase inhibitors.
Exactly. Those work well because, for one, receptors are often on the cell surface, making them relatively accessible to antibody drugs, and small molecule inhibitors can target the well-defined ATP binding pocket within kinases.
Okay, so accessibility is one issue. TFs are inside the nucleus, harder to reach.
Harder to reach, especially for large antibody drugs. But even for small molecules, there's a bigger problem: the nature of TFs themselves. Many TFs, particularly the parts that bind to DNA or interact with other proteins like coactivators, don't have the kind of deep, well-defined pockets that traditional small molecule drugs like to bind into.
They have flatter, broader interaction surfaces.
Often, yes. Their DNA binding domains recognize specific sequences, but the overall interaction surface can be quite large and shallow. And the surfaces they use to recruit coactivators can also be tricky. This makes it incredibly difficult to design a small molecule drug that binds tightly and specifically only to the TF you want to inhibit, without also hitting lots of other unrelated proteins with similar-looking surfaces.
The off-target effects problem. Toxicity.
Exactly. You end up with drugs that might hit the target TF, but also interfere with dozens of other essential cellular processes, leading to unacceptable side effects. Targeting TFs directly with traditional inhibitors has historically been very challenging due to this specificity issue.
So it's an accessibility problem and a specificity problem.
Precisely. But this challenge hasn't stopped researchers. It's actually spurred incredible innovation and driven the field towards alternative strategies.
Okay, like what if you can't block the TF's active site easily?
Well, one approach gaining traction is developing protein-protein interaction, PPI, inhibitors. Instead of trying to jam the TF's DNA binding site, these molecules aim to block the connection between the TF and its essential partners, like those coactivators or chromatin remodelers we talked about.
So you're not targeting the TF's main job directly, but you're preventing it from recruiting its necessary construction crew.
Exactly. If the TF can't recruit its team, it can't effectively activate transcription, even if it can still bind DNA. It's a more subtle, potentially more specific way to interfere.
Interesting. What else?
Another really exciting area is targeted protein degradation. Technologies like ProOTACs, that stands for proteolysis targeting chimeras.
Protacs. Okay, what do they do?
These are really clever molecules. A ProTac is like a molecular handcuff with two ends. One end is designed to bind specifically to the target protein you want to get rid of, say, our problematic TF. The other end is designed to grab onto a component of the cell's own ubiquitin proteasome system, the machinery that normally tags proteins for destruction.
So it physically links the bad TF to the cell's garbage disposal system.
Precisely. It forces the cell's own destruction machinery to recognize and degrade the target TF, even if the cell wouldn't normally do so. You're not inhibiting the TF, you're just telling the cell, hey, destroy this specific protein right now.
Wow. That sounds like hijacking the Wnt pathway's degradation trick, but using it therapeutically.
It's exactly that kind of principle. Leveraging the cell's natural disposal systems. It's a way to eliminate the problematic protein entirely, rather than just trying to block one of its functions. This approach holds immense promise for tackling traditionally undruggable targets, like many transcription factors.
So the difficulty in directly drugging TFs is just force scientists to get smarter, finding these clever workarounds, interfering with their partners, or just marking them for complete destruction, controlling the command center by manipulating its lifespan or its support staff.
It absolutely has. It's driving real innovation. And just to loop back to our initial point about cell fate, remember that many normal cells require continuous input signals just to stay alive. If those survival signals disappear,
the TFs conveying the stay alive message get deactivated.
Right. And often the default program kicks in the TFs promoting apoptosis get the upper hand, and the cell initiates its own destruction. So understanding TF regulation isn't just about growth or differentiation, it's quite literally about the cell's final decision between life and death. So just to recap the main ideas, we've really seen two fundamental ways TFs get activated. There's that rapid, direct route for lipid-soluble signals, like steroid hormones, where the signal binds directly to the receptor, which is the TF, often inside the cell already.
The express lane.
The express lane, yeah. And then there's the more common complex relay race for signals that can't cross the membrane. They hit surface receptors, kick off these intricate phosphorylation cascades involving kinases, phosphatases, and those crucial adapter proteins.
Building the specific signaling hubs.
Exactly. Building hubs, ensuring specificity. And the ultimate goal of that cascade is to activate a latent TF, control its stability, get it into the nucleus, and allow it to bind DNA.
And once it's there,
the core takeaway is that the TF is the final step. It translates that incoming signal into a durable physical change by recruiting the whole team, co-activators, chromatin remodelers, to reshape the chromatin landscape, and, most importantly, recruit RNA polymerase to drive gene expression.
So what does this all mean for you, the listener? Hopefully, you now have a much clearer picture that really everything from how your body responds to hormones, to how tissues develop, and even how devastating diseases like cancer arise. It ultimately comes down to these small modular proteins, transcription factors, receiving their activation signal, getting their PTM access pass, recruiting their team, and making the critical decision about where and when to bind DNA inside the nucleus.
They are the interpreters, turning biochemistry into actual cell fate.
And if we think back to that hubs and nodes idea from systems biology, the sheer complexity inside the cell is just staggering, isn't it? We talked about how one tiny change, one phosphorylation event, can dictate whether a TF lives or dies, moves or stays put, binds DNA or doesn't.
The computational power needed to manage thousands of these interactions simultaneously with such precision. It's truly mind-boggling. We're only just beginning to really appreciate the full scale of it.
Right. So here's a final, maybe provocative thought to leave you with. We've established that targeting TFs directly is hard because of specificity. And we've seen that targeting the upstream receptors works, but maybe isn't always the ultimate solution. We also talked about those adapter proteins, the ones with specific SH2 or SH3 domains that build the signaling hubs and dictate which pathway gets activated downstream.
The connectors.
The connectors. Now, what if the next big leap in therapeutics isn't just about blocking receptors or even degrading TFs with pro-attacks? What if it involves designing artificial adapter proteins?
Interesting. Go on.
Imagine synthetic molecules that don't just block a signal, but actively rewire it. Molecules designed to, say, bind to an activated kinase. But instead of letting it phosphorylate its usual pro-cancer target, they physically redirect it to phosphorylate and activate something beneficial, like maybe TP53.
So hijacking the internal hubs reprogramming the cell's wiring diagram on the fly to switch the output from a go signal to a stop signal.
Exactly. Could we engineer molecules that change the outcome of the signaling cascade rather than just trying to turn the whole thing off at the source? It moves the therapeutic intervention from the cell surface deep inside right into the network logic itself.
That's a fascinating concept, shifting from simple inhibition to active network engineering. It certainly opens up a whole new way of thinking about how we might correct pathological signaling in the future. A true deep dive into reprogramming the cell's command center.
Definitely something to think about. Thank you for diving deep with us today into the world of transcription factors.